Good morning, everyone. Thank you for joining us for another Immutep investor call to discuss more clinical results from the company's lead product candidate FD-Lugimod Alpha. Clive Tompkins in Sydney here, your moderator for today. At this time, all participants are in listen-only mode. We will conduct a question and answer session at the end of the presentation. I'd now like to hand the call over to Mark Vogt, Immutep's CEO, to start the presentation. Mark, are you there?
Yeah, thank you, Clive, and welcome everyone. Early in the morning in Australia, obviously, late in the afternoon in the US, and close to midnight in Europe. So we would like to thank all of you for joining us on the call today. And I hope that everyone is keeping safe and well with your families during the COVID-19 situation, obviously still impacting our lives. On today's webcast, we are excited to take you through the new data from our TACTI-002 and the Untitled trial, which was just announced at the virtual ESCO conference. The data relates to our lead product candidate, FD-Lugimod Alpha, also known as IMP321, or as you will hear us calling it, FD. Joining me on the call today from the company is Professor Frédéric Triebel, who is our Chief Medical and Chief Scientific Officer. And we are also joined by Christian Müller, our Director, Clinical Development and Regulatory Affairs. Now, the disclaimer, the forward-looking statement. So, I think you know practically our portfolio. We have two broad effects with LAG-3. On the one hand side, immunostimulation. On the left-hand side, where we try to increase the body's own immune response, or immunosuppression on the right-hand side, where we aim to decrease that response. And across these two categories, Immutep has 4 different product candidates, 2 in immuno-oncology and 2 in autoimmune diseases. 2 of these products are exclusively worldwide out-licensed to GlaxoSmithKline and Novartis, 1 in autoimmune diseases, 1 in immuno-oncology. And in our webcast today, we will focus on FD-Lugimod Alpha on the red box on the top left-hand side. FD is a unique antigen-presenting cell activator, or APC activator. So we use here LAG-3 as a tool to activate the dendritic cells via the target MHC class II. Now let's go into a little bit more detail. First of all, here the pipeline chart. You see that we have been putting together a diverse program of clinical trials for FD-Lugimod. There are, of course, also other trials which are run by our partners, but we are here concentrating really on the trials we are running. And our largest study is APEC. On top of that page, we reported progression-free survival results from APEC by end of March and expect overall survival data later this year. Our other phase 2 trial is TACTI-002, and interim data from TACTI-002 has been very encouraging to date, and we will go into more detail during today's session. FD is also being evaluated in different other solid tumors through our INSIGHT-004 trial, which is conducted with Pfizer and German Merck, TACTI-002 with Merck US. And we will be taking you also through the first data we presented at ESCO from that clinical trial. Earlier, we performed also a trial with anti-PD-1 in melanoma. That trial is called TACTIMEL, not in focus today. Here again, an important reminder. On slide 7, there's a diagram of FD showing its structure on the top left-hand corner of this slide. FD is a soluble recombinant fusion protein, and as mentioned, it's an APC, antigen-presenting cell activator. Its mechanism of action is unique, and it's first in class as an MHC class II agonist, which strengthens its value for Immutep significantly. It's important to note that FD is not a blocking antibody, like you see on the right-hand side of that page, but a unique antigen-presenting cell activator. So on the right-hand side, you see the classical immune checkpoint inhibitor here, anti-LAG-3. This is a mechanism often referred to as releasing the brakes on the T cell. This is definitely not how FT works, but the partnered program with Novartis. So FT is on the left-hand side, where we use LAG-3 as a tool to activate the antigen-presenting cells via MHC class 2. So target is MHC class 2, and this leads via the physiological way to an increased CD4, CD8, NK cells. So we push the gas of the immune system. And I would now like to hand over to our CSO and CMO, Frédéric. Frédéric, as you all know, discovered LAG-3 years ago and is the pioneer in the field and a rock star in the immunotherapy space. So he will go into more detail in the next slides regarding the data. Frédéric?
Mark, we'll now talk about the phase 1 trial, Insight-004, which combined FT with avelumab anti-PD-L1 therapy. Turning to slide 9, our Insight-004 study is a dose escalation trial of FT in combination with avelumab. We are conducting the trial in Germany in collaboration with Pfizer and Merck KGaA, and is being run under the protocol of the wider Insight trial by IKF, the clinical center. Insight-004 has 2 cohorts of 6 patients with solid tumors participating. Patients' recruitment was completed last month. Participants are receiving either 6 mg or 30 mg of FT in the combination therapy every 2 weeks for 6 months only, as it is a phase 1 trial. There is then a 6-month follow-up period where patients receive just avelumab. Now on slide 10, we are pleased to report that the combination treatment is safe and well tolerated by patients. There were no dose-limiting toxicity and no new safety signals with standard dose of avelumab. In terms of adverse reactions, FT has only been associated with 1 so far, and that was an injection site reaction. 3 patients out of the 12 have been treated recently and have therefore not been staged yet. At the present time, we have seen 4 partial response, 1 in colorectal adenocarcinoma, 1 in pleural mesothelioma, which is an indication not known to be ICI sensitive. A 3rd partial response is in squamous anal cell carcinoma, and a 4th one in gastroesophageal junction adenocarcinoma. Again, 2 indications not being ICI sensitive. We'll now talk about our other phase 2 trial, TACTI-002, which combines FT with an approved anti-PD-L1 therapy. Slide 12 gives you an overview of the trial design for our ongoing phase 2 TACTI-002, which we are conducting in collaboration with Merck Sharp and Dohme, known as MSD. TACTI-002 is testing the combination of FT with MSD drug Keytruda, which is an anti-PD-1 therapy, also known as pembrolizumab, in up to 109 patients. The study is a multinational open-label trial with a Simon 2-stage design. The advantage of this adaptive design is that it enables us to expand the cohorts when a predefined number of tumor response to the combination therapy are observed. TACTI-02 is a PD-L1 all-comer study, meaning that patients could participate without being pre-screened for PD-L1 expression at the tumor site. PD-L1 status is a well-known predictive marker for response to pembrolizumab monotherapy, and it will be measured centrally later on. We have 3 types of cancer patients participating in the trial. In part A, we have patients with advanced metastatic non-small cell lung carcinoma, and these patients are treatment naive, so they haven't received any previous treatments for their metastatic disease. Part B enrolled patients with advanced non-small cell lung carcinoma who have progressed on PD-1 or PD-L1 therapy, which has been shown on 2 consecutive CT scans. So this is a real refractory setting. And Part C, patients who have recurrent metastatic head and neck squamous cell carcinoma after failure of platinum-based therapy. The primary endpoint for TACTI-002 is overall response rate, or IORR. The endpoint is measured by iRECIST, which is the accepted standard for measuring responses in trials involving only immunotherapies. We are pleased that recruitment has progressed for the trial, even in the current environment, where COVID-19 is generally slowing patients' participation in clinical trials across the globe. However, we now anticipate that recruitment may slow down TACTI-02 going forward due to the closure of hospitals in certain countries that have been most affected, such as Spain or the United Kingdom. The most up-to-date recruitment numbers are detailed on the slides, and you will be aware that we have already been able to expand to stage 2 or 2 parts of the trial under the adaptive design due to encouraging overall response rate in Part A and Part C. Lastly, we should mention that the trial is taking place across Europe, Australia, and the US, where FT received its first IND approval last year. Moving now to slide 13. Here we are looking at interim safety results from stage 1 of all 3 parts. These data reflects results from the 76 patients who were enrolled in the study at the time of the data cutoff, which was 30 April 2020. The results are continuing to build FT very favorable safety profile, and no new safety signals have been observed in combination with pembrolizumab. Just over 40% of patients had 1 or more adverse events that were grade 3 or higher. There have been no treatment-related deaths. Overall, we continue to be very happy with safety profile reported from FT. You can see how this strong safety profile compares to other treatment regimens in the table on the right-hand side. In the combination with pembro, we do not have any treatment-related deaths, and we only have 4% adverse event leading to discontinuation. This bodes very well for patients and for our engagement with the regulatory bodies. I would now like to hand over to Christian Müller to take you through the unmet medical need and efficacy results around this trial. Christian?
Frederick, let's start with an introduction to first-line non-small cell lung cancer, where almost 2 million new cases are reported every year, as you can see here on this slide. Despite recent improvements, actually, for instance, with checkpoint therapy, there is still a high unmet medical need, especially for patients with a low PD-L1 expression, who account for approximately 70% of the total population. And I believe all of us are aware that efficacious treatments like pembro plus chemo have quite some side effects and also carry a huge burden for the healthcare systems. Nevertheless, this indication is one of the biggest markets in oncology, and I would like to show you some very encouraging data in the next couple of minutes. Looking at slide 15 now, the slide outlines the baseline characteristics of patients in stage 1 of Part A. These are patients in the first cohort of Part A with first-line non-small cell lung cancer. As Frederick has outlined, this is a PD-L1 all-comer trial. The table on the left gives you an overview of the patients participating in this part of the trial. As you can see, the median ages was 65, predominantly male patients participated, and approximately 71% had an ECOG performance status of 0. The vast majority of patients were current or former smokers, and we had almost equal parts of squamous or non-squamous histology. More importantly, as Frederick outlined, PD-L1 expression is a predictive marker for pembrolizumab monotherapy. It is important for us and also for the interpretation of the results to confirm that we do not have an overrepresentation of high PD-L1 expressors, meaning of a PD-L1 expression of 50% or more. As you can see, in this trial, approximately 31% of the patients in this part had a high PD-L1 expression compared to approximately 30% you would expect based on all the data out there in different trials, which is pretty consistent actually. So we believe that the data is pretty representative for the first-line non-small cell lung cancer patient population. Turning now to the efficacy results of this patient population on slide 16. We are delighted to report an overall response rate of 53% according to iRECIST and a disease control rate of 82%. In the waterfall plot on the right, you can see that there are responses among all subgroups of PD-L1 expression. The percentages on each of the bars are indicating the PD-L1 expression. The responders have the green color, and you can see it ranges from about 35 to approximately 75%, so pretty deep responses. Underneath the waterfall plot, we have detailed the PD-L1 expression in a table to show that we have responders basically in every PD-L1 subgroup. One out of three patients with low PD-L1 expression or no PD-L1 expression basically had a response. 3 out of 6 with a medium PD-L1 expression and 3 out of 4 with a high PD-L1 expression. And then for 4 patients, the PD-L1 levels were not evaluable in the tumor specimen. And there we had another 2 responses. We also could show in the data that, or we could also show in this part of the trial that we had responses in the non-squamous part and in the squamous part of patients, which is also very important if you think ahead of potential registrational trials. Looking now at slide 17, on this slide we illustrate the individual patient responses over time through a spider plot. Each colored line represents one patient and the development of the target lesions over time. The x-axis is in weeks and percentage change on the y-axis. The color of the line indicates the PD-L1 expression category they belong to. Red means this patient has a PD-L1 expression of less than 1%, orange, PD-L1 expression 1 to 49%, and green indicates a PD-L1 expression of 50% or larger, and black are the patients where the PD-L1 expression was not evaluable. The lines with an arrow indicate that this patient is still continuing with the combination treatment. As you can see, we have 7 patients, which is 41%, still under treatment, and the vast majority of them are almost under therapy for almost a year, which is, as I will show you in a minute, quite long for this patient population. The median progression-free survival is estimated to be 9+ months, and as I just mentioned, generally for this kind of patient population to have like 9 months or more than 9 months median progression-free survival and an overall response rate of 53% is really, really encouraging, given that this is really a PD-L1 all-comer trial. We will take a moment to reflect on that on the next slides to show what it means in the context to other available therapies. As you are aware, there are different approved and tested options for these patients. Each of them, as we believe, has different drawbacks. The doublet chemotherapy for long, the standard of care, has a pretty low overall response rate of 20 to 30% and a short PFS and OS and is relatively toxic. Pembo plus chemo has certainly improved PFS, OS, and overall response rate, but comes at a price of substantial toxicity. And as you can see, it has a shorter duration of response compared to ICI products alone. EP plus Nivo, an ICI combination, is slightly better than the doublet chemo, but also carries some drawbacks in terms of toxicity and cost for the healthcare system. Pembo alone is efficacious in high PD-L1 expressors, but for patients with medium, meaning 1 to 49%, or low, less than 1%, overall response rate is 17% or less. And I think very important to note is that Pembo has never been officially tested in large trials in PD-L1 negative patients, which is allowed in our protocol. Based on the results we have presented today, FD offers a response rate, clearly higher response rate than Pembo alone, despite allowing for PD-L1 negative patients and without adding substantial additional toxicity. Overall, we believe FD offers a compelling potential treatment option in first-line non-small cell lung cancer and a unique opportunity for differentiation for big pharma for their PD-1 and PD-L1 therapies. Just to remind everyone here, as of now, there are 3 PD-1, PD-L1 therapies approved for this indication. This is Pembo, Nivo, and atezolizumab. And probably there are more to come. So big pharma will need a differentiator here. It's at least our belief. Moving to slide number 19, we would like to touch base on the epidemiology of squamous head and neck cancer for a moment, as this is part C of our trial, and also quite an interesting indication for drug development in oncology. There are over 500,000 people diagnosed with this disease each year, and approximately 350,000 develop metastatic disease. Despite all recent improvements, especially with PD-1 in first and second line setting, stakeholders like patients, doctors, payers have the same unmet need as for first line NSCLC, only more dramatic, as the response rate, as we will see in a second, and the overall survival rates are considerably shorter and lower than for non-small cell lung cancer. We just picked one example here. There's a Kaplan-Meier plot of the progression-free survival from the KEYNOTE-40 study. Second line head and neck, same indication basically we are targeting in our trial. As you can see, at 5 months, approximately 30% or less are still progression-free. So this disease is really, really aggressive, and patient and other stakeholders really want improved treatments, and there is a need, a high unmet medical need here for efficacious treatments. Moving now to the results from the second line head and neck squamous cell carcinoma patients, which are on slide 20. Patients are all pre-treated with platinum-based chemotherapy, and about 47% have an ECOG of 1. Median age is 66 years, and 94% are male. Here we detail the patient responses at the data cutoff date in the table on the left-hand side. We are very encouraged to report that the data has evolved positively since the last readout for AACR, which, just to remind everyone, was not long ago. We now have staged all patients after treatment start at least once, and we observe an overall response rate of 39%. This is an improvement to the overall response rate reported in March, which was just about 33%. As you can see in the waterfall plot on the right side, the responses are getting deeper, and we are very happy to announce that we now also have one complete responder, meaning that all target lesions have disappeared in this patient, who is indicated by the blue bar, and partial responses as for the non-small cell lung cancer part are indicated by the green bars. Taking a look at the spider plot for this group, let's turn to slide 21 now. You can see the patient with a complete response is again indicated by blue and had a 100% shrinkage of their tumor target lesions at week 36. It's the most recent staging for this patient. So this patient, as you can see, indicated by the arrow, is still ongoing and still under treatment. Besides this complete response, there are 2 other interesting cases here. There's one patient with a pseudo-progression first and who responded later on. And then there's one quite late response at 8 to 9 months. In total, 8 patients are continuing, as of data cutoff, the treatment indicated by the black arrows. And You may recall the PFS plot was shown on slide 19, where about 30% only were treated at 5 months or thereafter. So taking this all together, these results are quite remarkable. They're pretty early on, but making us very optimistic for the combination of FD and Pembo in this patient population. On slide 22, we would like to do the same exercise as we did for the non-small cell lung cancer part to show you some comparable results in second-line head and neck to put context to our data. As you can see, patients are treated with chemo or Pembo as monotherapies, have modest overall response rate. All of them are less than 18%, and the median progression-free survival is just about 2 months. In this context, we believe FD's performance combined with Pembo is remarkable. We are seeing a 39% response rate, which is clearly higher than what you would expect from Pembo alone. And as for the non-small cell lung cancer part, it doesn't come along with substantial additional toxicity, as Frederick mentioned earlier on. So overall, this makes it very exciting, potentially treatment option for patients and physicians, given the disease is so aggressive. The combination therapy also potentially holds some shortcuts on its way to registration. And here I would like to give some more background thoughts on that. And I would like to show you one example on the next slide, how Pembo was approved as a second-line treatment by the FDA in 2016. The approval came based on non-randomized studies, based mostly on the KEYNOTE-12, which is an open-label multi-cohort phase 1b study. And here they achieved an overall response rate of about 16% with Pembo alone, median PFS of 2.1 months, and the median duration of response was not reached at the time. They submitted it, but it was shown that it was quite durable for at least 23 of these 28 patients who had a response here, with a duration of more than 6 months. The FDA put a condition here. They required a phase 3 trial, the KEYNOTE-40, which failed later on to show significant improvement in median overall survival. But the label was not changed, and to be fair, they just missed it by a margin. Given the pathway and the approval outcome for Pembo, FD shows promising activity based on the initial results from TACTI-002 and 2 very interesting indications in metastatic cancer. And Mark will now provide some concluding remarks for our webcast, and I would like to thank for your attention so far. Mark.
Yeah, thank you, Christian. This brings us to the end of the newest TACTI-002 results. So let's come briefly to the summary and outlook. First of all, here on slide number 25, I would like to remind everyone that we have now, I believe, a good experience with the anti-PD-1 or anti-PD-L1 combination. So FD is being evaluated in up to 145 patients in different cancer indications, from non-small cell lung cancer, head and neck cancer, we just learned about that, to melanoma. Let's not forget about TACTIMEL, where we presented final efficacy results in October last year, and other solid tumors via Insight 004. So there's a growing pool of efficacy data we are gathering in each indication. And interestingly, I believe that for the time being, it was quite nice to see also a kind of a consistent, good pattern, good hints of activity of the combination of eftilagimod with anti-PD-1 and anti-PD-L1. We left out the scientific rationale during this call. We discussed it in the past, and we are quite glad that the data keeps evolving here. So what could be next in general, just as we have or had ESCO, there were many different options and therapies discussed. I believe FD is emerging really as a strong potential therapeutic option in the IO landscape. And the results from TACTI-002 and other trials are very encouraging and compare well to what has been approved so far. So I believe there might be a role if the data continues to be as strong as it was. And Also not to forget that we have two different immuno-oncology product candidates, and one is already out-licensed to Novartis. So this puts us in a good position when it comes to the potential next big thing in oncology. Of course, it's all in a very data-driven mode, and there are other good approaches. Other data is out there. So there are other agonists, antagonists in immuno-oncology that are being discussed. For instance, at this ESCO, TIGIT was a bit in the focus. And I believe that we and our partners are well positioned here. And we should also not forget, in terms of the LAG-3 antagonist, so anti-LAG-3, about Bristol Myers Squibb. I think they mentioned in one of the conferences 2 weeks ago or so that they expect phase 3 data with their anti-LAG-3 in melanoma either end of this or beginning of next year. Of course, they have to report that, but for us, it's very important if LAG-3 data is reported. So this brings us to the end of the overview, just the outlook very briefly. We have more things to come during this year, so more data. Potentially, the next stop might be ESMO in September for TACTI-002. Also, I believe there will be further data updates from the Pfizer trial, so from INSIGHT-004. In metastatic breast cancer, overall survival should be due by end of this year. And of course, we will report also the progress from our partners, GSK or Novartis. And we hope also, of course, for regulatory interactions and the results. So this brings us now to the complete end of the formal part of the webcast. I would like to thank Frederick and Christian for their presentations and their contribution here. And I now will pass back to Clive for questions from the analysts on the line.
Okay, thanks, Mark. And look, just a reminder to the analysts, there's a raise hand icon at the bottom right-hand side of your screen. If you'd like to ask a question, please click on that. Our first question comes from Tanushree Jain at Bell Potter. Tanushree, can you go ahead, please?
Thanks for taking my questions, and congratulations, Mark, Frederick, and Christian, on such an encouraging set of data. My first question is around the PFS for both the Part A and Part C results from TACTI-002. So we've got an estimated PFS of 9+ months for Part A. Can you confirm that data cleaning is the reason we haven't locked that in finally?
So I hand this over to Christian.
Thanks for the question. There are different reasons for that. So the main reason is that we haven't had seen enough events, and then yes, certainly, as you say, we're about to clean the data, and don't want to correct it later on. That's the most reasons, and you can expect to see that later at ESMO.
Right, and just on the Part C, in terms of the patients who are still on treatment, it looks like we should have reached the median PFS there as well. Can you give any comments on that?
It has not been read out yet.
But we are on it. Just to add to that, we are on it. You are right. We just want to report data where we are 100% certain. I think it's fair to say that the PFS is tracking well also for Part C, but we don't want to state a number yet because the data should be fully cleaned for that part as well.
Right. And then Mark, just on the landscape, and I guess some of the other presentations in non-small cell lung cancer, including the TIGIT, which came out at ASCO, it looks like the chemo plus pembro combination in first-line non-small cell lung cancer is really unchallenged at the moment. So given that backdrop, I guess from the FT pembro combination, the results that you're seeing, can you perhaps give some strategic direction as to whether you could perhaps help Merck maintain that leadership position, or do you think some of the other contenders could benefit from combining with FT, should results continue in this direction?
Yeah, thank you. I believe, first of all, it's the combination with pembrolizumab, but the principle should apply to other anti-PD-1s as well, anti-PD-L1 maybe also. You are right. The pembro-chemo combination, also from our point of view, if you look at some of the data, is not really challenged. The TIGIT results have been nice, I believe, as one saw in a randomized setting a difference, even though that difference or the absolute results were maybe not challenging pembro really, or pembro plus chemo. Eftilagimod, if the data continues to be that strong, could definitely be an option. Of course, there are then always questions how to address this in larger clinical settings. Also, one can think possibly also of a triple combination of eftilagimod, pembrolizumab, and chemo. So we believe, given the results we see today, to date, we definitely have a chance to improve the situation in first-line non-small cell lung cancer.
Great. And just on the head and neck cancer, when do you expect we'll get in response by PD-L1 expression data on that?
I guess that will also be at ESMO if we have a chance to present there. Don't forget that the PD-L1 expression measurement is done under the control of Merck. So there we are receiving the data from them.
Right. And just lastly from me, Christian, you mentioned the faster pathway in head and neck cancer. And I guess given the low survival rates there, I think there is definitely a more urgent unmet need there compared to non-small cell lung cancer. Some of the examples that you did put up, they obviously had much larger set of patient population, like 174, etc. So can you perhaps talk about what could be the fast-track pathway from here? I'm assuming you will still have to do a pivotal trial, but do you expect that to be more in terms of patient numbers similar to what you've seen previously with Keytruda?
Christian?
Thanks for the question, a very important one, actually. So first of all, we believe that the data we have so far in the stage 1 looks really, really encouraging. And we will take the data as soon as we have a bit of more flesh on the bones to the regulatory agencies and discuss with them. And we believe, based on the current landscape in head and neck and based on what has been seen in the past by the actions taken by the regulators and the options they have been given to other companies, that in this indication, as there's such a high unmet medical need, you may have a chance with a non-randomized trial. The patient numbers and the actual design certainly needs to be discussed with the regulators in specific meetings. But as you say, it's certainly an indication where the unmet need is very high, which always plays a role for regulators.
Great. Thanks so much.
Okay. Next question from Dennis Holm. Dennis is at Taylor Collison. Dennis, go ahead, please.
Thanks very much. And congratulations to Mark and the team on the data so far. It's great to see the evidence for FDs continuing to build. And in relation to that, there's 2 cohorts of lung cancer patients which have almost completed recruitment. And I'm wondering, when can we expect to see data coming through from those 2 cohorts? And can you just talk about what's the rationale about the anticipated timing of disclosing that data?
Yeah, thank you, Dennis. So first of all, I believe that we have a chance, and this, of course, is always dependent how the patients are tracking, also under the COVID situation and the recruitment, of course. We have a chance to see data at ESMO. Again, that is mid of September. mid to end of September and will be entirely virtual again, as far as I'm aware. Please don't forget that we fully recruited the first cohort, so part 1 of stage A, in June last year, and then we presented first data at SITC, which was to a certain extent also immature. So unfortunately, it takes a while. We are very eager to put out data as soon as we can, but of course, it should be robust and should also then be big enough, so to speak, to make a good difference. So we are working on it. It's our interest to put it out. I believe we have a good chance to see it at the next big conference for us, which is ESMO.
Great, thank you. And then just moving across to the INSIGHT study, I've just been sort of tossing up what I should make of the INSIGHT 004 data. On the one hand, 44% response rate in evaluable patients is fantastic. On the other hand, 3 out of those 4 responders had tumour biomarkers that are typically associated with above average response rates, such as MSI high or high PD-L1 expression. So I guess, how should we think about that data and how it compares to what other products have achieved?
Yeah, first of all, I would say the aim of the trial is safety, and we have achieved that. It's a phase 1 exploratory character. One should not forget, with 2 different dosages of eftilagimod, 6 and 30 milligram. And we are actually very glad that we see activity here, especially as these patients typically do not have a treatment option. So typical phase 1, we see here hints of activity. I think it's worthwhile to look on a single case basis, as we have a variety of different tumors. And as Frédéric pointed out, and he may want to add to that in a second, some of these patients are not typically immune checkpoint inhibitor sensitive, whatever it means in at this early stage. But we have been very encouraged, and I believe in general it's nice to see. But Frédéric, you may want to go more into detail.
Yes, I mean, these patients have been treated for only 6 months, as it is a phase 1, not for a year, we usually do for phase 2. And indeed, we have seen 2 late response in part A of TACTI-002 and a third one in part C, patients responding after 8 months or 11 months. So it's difficult to say at the moment, but some of these patients responding to the combination have been treated for 1 or 2 lines. I mean, and there are some kind of niche indication and not shown to be responsive to anti-PD-1 and certainly not to avelumab. So it's difficult to say. These are 4 anecdotal cases, and out of the 12 patients, 3 have not been staged. So we may have other responses. We'll know more later on.
And I guess in relation to INSIGHT, what are the next steps after those last 3 patients have been evaluated? Would you look to expand any of the cohorts? Particularly, for example, if you got a second responder in colorectal cancer or gastroesophageal junction cancer?
So first of all, I would say from a safety point of view, and also, let's say, from anecdotal activity in this phase 1, also together then, of course, with the results from TACTIMEL and TACTI-002, there would be definitely the basis to further explore this combination. Of course, as you will understand, we have discussions around that, internal discussions also with the involved parties. So I don't want to make any public statement at this point in time.
Mm-hmm. And just changing tack, are you seeing much impact of COVID-19 on recruitment, and more particularly on patients having their follow-up tumour assessment scans?
So in terms of recruitment, I think we are in the good situation that on the one hand side, APEC fully recruited, that INSIGHT TACTI-004 fully recruited, the 12 patients, and that we have more than 70% recruitment of TACTI-002. Indeed, it's a little bit slower than it has been before. In terms of the scans and the assessments, it's we evaluate this on an ongoing basis. There might be an impact here or there, but I believe as we have a global clinical trial practically with Australia, which was not really interrupted by COVID-19 in terms of the hospitals, the US and Europe, we have here a relatively balanced approach. So any potential impact should be manageable.
Okay, that's great. And so I guess changing tack directly, but staying on COVID-19, can you touch on how the discussions about a potential study of FD in COVID-19 patients is progressing?
Yes, it's progressing. We have been approached by a PI to test FD also in the COVID setting. I mean, it's a good idea to strengthen the innate immune system, especially in order to prevent patients which are hospitalized, but not in the ICU from getting into the ICU. So let's say a little bit earlier intervention, especially in absence of a vaccine. You may know that we have also patent claims around that, and that the potential of eftilagimod in infectious diseases is actually in our slides since years, and also in the patent claims. We are fully focusing on oncology. We are happy to support the efforts against COVID, and we will inform the market once there is more to say.
Great. Well, thanks very much. That's all from me.
Okay, and Mark, we've got two more people asking questions. Next is Chris Redhead from Gertz Partners. Chris, would you like to go ahead?
Yeah, quick point on, I think, on the PD-1 and solid tumours. Quite a lot of those tumours, you get PD-1 expression, but doesn't give you a response, right? So it's not like quite a lot of those tumours don't do that. Just that point. The other point was chemotherapy, which I think is, so if you look at the APAC trial, you saw some effect with some synergy perhaps when there was chemo there, but not, it seemed to disappear when you took the chemo away. I'm just kind of wondering how that would roll into some sort of triple therapy, whether it, you'd have to sustain the chemo. How would you think that would work?
Yeah, first of all, I believe you're right in terms of your comments. And the second is that one should, of course, learn and continue then with the clinical trial design, meaning giving the combinations. Let's assume there would be a triple combination as long as it is feasible and justifiable. So one would definitely try to continue that then in larger settings. But we have not made any internal decision to go into the triple combination. We have, of course, a number of different parties we discuss with different things. Can't be more precise at this point in time. So there are plenty of ideas, and if we would move into that direction, then I think one would aim to give it longer together.
Okay. A quick thing with radiotherapy, and I think that if you look quite frequently, if you see a synergy between chemo and IO, then you get similar synergies between, or even stronger synergies with radiotherapy. Is that something that you're thinking about?
The potential of eftilagimod is very broad. So indeed, one could aim also to go with radiotherapy. Of course, we have to focus. We have to follow the signals we see. I mean, at the end of the day, we are treating the patient's immune system, not cancer directly. This opens up a great variety of different combinations. Our resources are, of course, limited. So this is why we are currently focusing on what we have presented in the past and today. And we'll continue to do that in the future. But if the setting is right, for instance, from a business development point of view, then, yeah, there are a variety of different other ideas.
So let me just wrap up. That's it. Yep. Look, no more questions today. Mark, can I hand back to you, please?
Yeah. Thanks a lot, Clive. And on behalf of the Immutep team, I would like to thank everyone for joining the webcast today. Of course, especially also the analyst, but also all others supporting us. Immutep continues to report encouraging data around FT, and we look very much forward to keeping you informed of further readouts. I mentioned already where the next potential stop could be. But I'm sure we have other things to say between now and September. A copy of the webinar will be available shortly on our website, Finance News Network website, and other media channels. Thank you very much and goodbye.